Yeah, I think we are ready to go. Rachel, do we have anything to say? I think we are good to go. Yeah, we have still some slots for this year, so I think next week, no, in 2 weeks, Sylvie Ruskin is going to talk about RNA structure prediction using experimental data. But then, yeah, if you want to talk this semester, let us know. And today, I'm really happy to have Quentin Vicens. So we collaborate at the moment. I don't know. Probably this is not. I don't have to disclose that, but I'm really excited about his work. So what he's trying to do in his lab right now is to do what we all kind of try to understand, how RNA shapes code for the functions, and also how to use small molecules to change functions and shapes of RNA. So he did his PhD in Strasbourg in France with our beloved to our community, Eric Westhoff, working on interactions of small molecules and ribosomes. Then he moved to Boulder, Colorado, when he worked as an assistant research professor with Jeff Keeves and Thomas Cech. And now he's building his own group at the University of Houston in Texas. So I don't know how much Texicana is being shown today, but yeah, as with the title, I think he's really excited about the place. So in his lab, he's exploring different techniques of biochemistry, molecular and cell biology, and also bioinformatics to understand more Z-RNA molecules. And I think what is Z-RNA we'll learn today more. And also to understand how to use these techniques for RNA structure determination and predictions, and also use that for targeted gene integration. And this I just checked and I learned about him. We talked about it briefly, but now he is also co-founder and head of biology at InnoFocus Therapeutics. So I think he tries to combine great teaching, apparently, what I read on the internet, also great science, and now trying to get some more money out of that. So the screen is yours, Quentin. I'm really happy.
All right. Thanks, Magnus. Thanks, Rachel, for the invitation. This is a great webinar series. I've been enjoying it. I've been recommending it to people, and I'm glad to have an opportunity to talk about the stuff that we do. So I did recently move to Houston. So my title is obviously a play on the words of the quite famous right now Beyoncé album that just came out. The references to that are multiple, but one of them is obviously because Beyoncé was born in Houston, as some of the faculty member who interviewed me made sure they would point out. But I want to talk about a lot today about Z-RNA. So the play on words is also about that because Z-RNA is a left-handed double helix that we look at, and obviously, opposite from the right-handed typical A form. So the way, we typically draw RNAs is like that, like shown on the left, right? Typical secondary structure with maybe some paired regions and some loops, bubbles that maybe indicate that these are unpaired. They may be not Watson-Crick paired. But when you look at the structure of this in 3 dimensions, everything is at least stacked, and maybe non-Watson-Crick pairs in motifs, Watson-Crick pairs in those gray regions. So really, RNA is this, as Eric, my PhD advisor and longtime mentor now, 30 years almost, RNA architectures are an assembly of modules, right? That's the way we think about it. Some of us talk about Lego pieces or tectons. So those motifs shown in colors here, right, are responsible for the cool stuff. They make a bend, right? For example, here in the RNA or here, they can participate in long-range interactions. We've heard a lot about these in that seminar series. They can bind other RNA. So for example, here, this is the T-box riboswitch, right, bound to a tRNA. So using interactions up here, down here, protein binds to that region in orange. So the A-form helices, right, often are seen as like, hey, I need spacers because I need a certain number of base pairs to go from that green hinge here to the region that's going to bind the DT loop of tRNA and so forth, right? So maybe they're kind of boring, right, to us. Like a lot of people focus on the Watson-Crick base pairs and, you know, siRNA and microRNAs. And in our field, we like to look at the non-Watson-Crick paired region usually, right? So today, I'm gonna focus on pieces of A-form helices that can also do some cool stuff because they can flip from right-handed to left-handed. And that's a work that started with DNA in the 70s with these two guys down there, Tom Jovin and Fritz Pohl. And they basically demonstrated using spectroscopic techniques that even though we had a double helix, you know, the B form from Watson and Crick that was right-handed, what they saw was under some conditions like high Sol, they would get left-handed conformation. And they demonstrate that by sitting on their desk, right? Where Tom Jovin's turning to the right and then Fritz Pohl turning to the left. What does that mean in terms of chirality? So this got crystallized in the same decade, actually, by Alex Rich from MIT. He worked on DNA and then several years later crystallized the RNA. You can see they superimpose pretty well, even though the starting A-form RNA and B-form DNA are not the same. And this is actually the first crystal structure of DNA ever solved. It's in the Z conformation. So when this came out in the late 70s, everybody thought they would see the Watson and Crick double helix, and they didn't, right? They saw that. So it started some kind of controversy, right? And when you ask Jim Watson, well, what do you think? Is that zDNA, does it even have a function? And Jim would say things like, well, you should ask Alex Rich because he's the only one who thinks there is one. So there's been this, you know, sort of stigma attached to these left-handed conformations, and people thought they're just like an oddity, an artifact. And fortunately, some people like Nacho Tinoco, who was the co-discoverer of the Z-RNA, said that, well, you know, it provides another example of the attitude of some biologists to supposedly non-biological results. So me and others are more of that mindset of thinking like, well, if we found that somewhere and we think it's not biologically relevant, maybe it's because we haven't looked hard enough. And we started working, you know, in like the mid, I think 2015 or so 2014 with Pascal Offinger down there, who also spent many years working with Eric Westhoff. And so if we look at this, you know, zDNA here, double helix on the left, it's really a succession of dinucleotide repeats. I mean, you need 6 dinucleotide repeats to make a full turn when you're in the Z conformation compared to like 11 when you're in A form. And when you look, you know, down the helical axis, yeah, you see a CG Watson-Crick pair with 3 hydrogen bonds. It's in cis, pretty much like in the double helix, the right-handed one. We looked at each of these steps, right? So it's a base pair, and here I'm looking at just like 1/2 of the base pair. So one of these steps, and traditionally it's been studied as CG, so pyrimidine, purine. And if you compare, so on the top, that CG step in zDNA compared to the same CG in bDNA, there are quite a few differences. So for example, in the Z step, the sugars, right, point in opposite direction, as you can see from this O4 prime. And that places the O4 prime of the first nucleotide in the step in this lone pair pi interaction with the ring of here, the guanine. And that guanine is in syn conformation, which, you know, is also a rare conformation of purines. And we have an alternating C2 prime endo, which is the favorite sugar conformation in DNA, and C3 prime endo, which is the favorite in RNA. So this is, you know, quite a concentration of rare nucleotide conformation within just 2 nucleotides, right? And we call that the Z step. And what we found with Pascal was that that Z step was found in several RNA families. And I'm showing a few examples here. We had a few more. And it was remarkable how this was conserved in those families and completely absent from others. So we saw the UNCG, for example, very famous tetraloop, very stable. The CG in that loop actually has Z-step conformation. When you look at HDV ribozymes, we see a Z-step at the catalytic site. I joined the Kieft lab at some point, as Magnus mentioned, and at that time, the Kieft lab had solved this tRNA-like structure from viruses at the 3' end of the genome. And there's a Z-step in that structure between the tRNA-like structure and an upstream pseudonode domain, so like domain interface. We also had example of the purine riboswitch, where we had the Z-step at the ligand binding site. So we had several hundred of those that we came across. And we also saw that the Z-steps, when we survey the databases of structures, are specifically recognized by proteins. So on the left is an example of a protein that makes specific contacts to it's actually here a UG step, UGZ step. That's important for T cell activation. The one on the right is more famous, and I'll talk a lot more about this one. It's involved this Z alpha domain from this enzyme called ADA1, adenosine deaminase acting on RNA. And that's the first protein domain that actually Alex Rich identified to specifically bind Z-RNA or Z-DNA or both. And this domain is found besides ADAR in other eukaryotic proteins like ZBP1. It's found in a PKR homologue in fish, and it's found in some viral proteins. So it's always found in proteins that have something to do with this immune response. And in the case of Z-RNA in a double helix, then the two sides can bind two Z-alpha domains. So the Z-steps that I showed traditionally studied were CG repeats. Well, we saw that, as had been seen with DNA, but also with RNA, they can be any sequence. Pyrimidine-purines, but also purine-purine, or pyrimidine-pyrimidines. So this really opened up, our questioning in terms of like, oh, this can really be anywhere. So what does that mean? So we looked, for example, at, we did a survey of ribosome structures. And, they always are treasure chest of three-dimensional RNA motifs. And showing here on the left is the secondary structure of the large subunit ribosomal RNA. And on dark gray here, if you see on the loops, these are all the U-turns that we find. So tetraloops with a U-turn, like shown on the right here, where it's the same. It's called the U-turn because it comes from the anticodon that has a U right before the anticodon. So it's a great way for a tetraloop to position three nucleotides, usually adenines, to make interactions in the minor groove of a helix, in a receptor. And we've seen that several examples of how this can be used in this seminar series. So there's several of those. And then we identified, so a UNCG, which has a Z-step, and then a G3AA that has the same sequence, right, at all these U-turns, but is also adopting the same conformation as a UNCG. And that's shown right here. So if I compare the structure of that UNCG to the one of that GAAA, they're pretty much the same, where we have, this full nucleotide in the loop. The second one is pointing away from you. And then the last two shown in red are forming the Z step, right? With that lone pair pi contact, the sugars in opposite direction, the last nucleotide in syn, et cetera, et cetera. And in both cases, all the ribosome structures that we saw, The second nucleotide in cyan here that points away from you is involved in a base triple. Okay, so it's not the same base triple for the UCG and that GAAA, but it's still involved in a base triple. And so the point being here is like, well, if we just look at the secondary structure and we see GAAA, we're going to be like, okay, it's a U-turn, when it's not a U-turn in the actual structures. So this is actually what it looks like, because these two loops, on top of being, you know, cool because of the Z step, well, they interact with the same subdomain 4 four-way junction as what we call this sort of complex double Z-turn receptor. So this is not a simple receptor in a helix with a certain number of defined nucleotides that we could easily plug and play with, put it in other contexts, so that we would even see in other contexts. We had a couple other examples that would support that. But since there was a work we published here in like 2018 now, but since then, I mean, we've looked at some of the structure that came out for this pre-ribosome complexes, like the states during ribosome assembly. And actually, we saw that GAAA is actually a U-turn in earlier stages. So there's some stages, right? Some transition that has to occur during ribosome maturation for the GAA to flip from the U-turn into that Z-turn, right? So it makes these, you know, transition from something that's going to be more A form-like to Z form-like quite biologically relevant. So the bottom line now at this point is that, you know, Z-RNA results from this combination of rare conformations. Like that's, we narrow this down to this dinucleotide we call the Z step. Immune response proteins recognize the Z step. I show some example of that. We found these steps outside, right, of these double helical contexts. And I think it's clear, at least I showed an example here with the ribosome, for example, and relevance into ribosome assembly, is that studying these Z steps is key for RNA biology. So when I joined Colorado, got back to Colorado in 2017 or so, right before moving to Houston, I was keen on like, okay, well, we need to find some experimental ways, systems for studying this Z formation. So how is the Z-RNA stabilized? So we know that if we increase salt, you know, 4 molar, 6 molar, depending on the salt, the sequence and all that, we can stabilize that left-handed conformation. We can also have some chemical modifications that actually decrease the salt requirement for flipping to Z. And we can have the proteins, right? I show a zoomed in version. Here's the zoomed out version of a piece of left-handed RNA bound to 2 of these protein domains that stabilize it. So we, together with this fantastic team led by Beat Vögeli here at Anschutz, and including Parker, Jeff Kroll, 2 PhD student, as well as Mokos, who is assistant research professor, as well as Rob Welty, current facility manager of the biophysics facility, Sean Beavers, the previous one. Connor was a PhD student who rotated with Beat and extended his rotation and kept working on this NMR project with us that was Z-related, and Lily Beck, latest edition. So together with this fantastic team, we sort of developed a pipeline, right, of biophysical strategies for studying Z-RNA, Z-DNA, making sense of this. We're all believers that this is real. So 2 examples here where we use, for example, circular dichroism on the left, because that's traditionally a spectroscopic technique that's really good for looking at chirality. So if we look from something that's A form, that gives us a peak here at about 260 nanometers, and that shifts to a peak at 285 or so in presence of high salt, then we have a good way to monitor. And if you have the protein, for example, here, that Z alpha, we see that shift. So it's indicating we have a Z form bound to the protein. When we use point mutants, we're interested in that are disease related and suspected they were not stabilizing the Z. That's exactly what we saw, right? We're not flipping now to Z. And on the right, we also have, so Beat's expertise and his lab are NMR, which is great for looking at these dynamic systems. So here we have titration, right? Increasing from bottom to top, the protein, looking at signal on the RNA, and we see peaks disappearing. Those red arrows as we increase the protein, indicating destabilization, and then some line broadening at the top, those green arrows indicating binding. So we have a toolbox, right, that we applied to various systems, and we described this at length in this various papers over the years. So we showed early on that when we took some RNAs that had been predicted by others as like targets for that Z-alpha domain in the transcriptome, including this alufold back here. So imagine this piece within the 3' UTR of an mRNA, so something really long. And we just took a piece of it and looked at that. We saw Z-alpha binding to some regions that are not necessarily CG repeat. There's one here, UG on the other side, and flanked by some regions that were not in Z, but destabilized. And we did that for a collection of fragments that we were interested in for various reasons, including stem loops. And so we sort of see that pattern, right, where we have like a section that's in Z some of the time, and then next to a destabilized Watson Crick paired region, mostly. And so not the entire fragments, right, adopts the Z-RNA conformation. So we developed this model of thinking, right, where we like, okay, we have this maybe long stretches of double-stranded regions. We don't know how long, we don't know which RNAs exactly. We're working on that. We have some hints, right, that this would be some of these foldbacks. We also, other people have shown that, oh, these RNA viruses, right, that infect cells make double-stranded RNAs as well when they replicate inside the cell, would have long stretches of double-stranded regions. And within that, we have some Z-prone regions, as we like to call them, right next to this less stable A-form region. And upon presence of Z-alpha, this leads to dynamic formation of this Z-form, right, within a context of A-form region. So we don't know what goes on exactly at the junction between A and Z. That's also something we're interested in. So one of the next questions we asked were like, well, how does the flip to Z happen exactly? Because we have to go right from a right-handed to a left-handed, and I show all these rare conformations that the Z adopts. So we used most recently control, that's locked nucleic acid, as a proxy for like something that's in a form that we know is not going to flip. So we show that it's not going to flip because it has this covalent link, right, between the 2' hydroxyl and the C4' of the sugar. So it adopts, more or less, people have crystallized it, something that looks like an A form. And we saw that the Z-alpha domain that's, stabilizing the Z also binds to the A form with a, dissociation constant that's not that bad, honestly, whether you look at it by NMR or by ITC. And it's about the same as an RRM binding to an RNA, single-strand RNA. It's about 10-fold less than to the Z-RNA, which is more like in the hundreds of nanomolar. And we also showed that there's multiple binding sites. So it's, and we showed also, that's a paper that just came out, that the surface of Z-alpha that interacts with this A form is the same as the one that binds to the Z form, as shown here by this, colored, the more red-orange, the more shift you see when you compare the end-bound to the bound. So we start with this study depicting this cartoon model, right? This pathway going from A or B form to Z. So one step is we know that there's some kind of sampling, right, of the A form by the protein domain. Another question was like, well, is there a Z state within the population of A? So this is what this, convoluted experiment, NMR experiment does. Very good at doing is pinpointing minor states, similar to experiments done by Alashimi at some point, if you're familiar with that, on DNA. Looking at, okay, under certain conditions, we're going to look at pick up on some minor states. So how do we do that? How do we detect this invisible state using this R1-row experiment, which looks at microsecond to millisecond time scale. So you should be able to detect something as low as 0.5%. So if we had 0.5% of Z within an RNA population, at high concentration for NMR, that's mostly A form, we should be able to see that. And we don't see that at 25 degrees, like we have a flat line, right? I'm not an NMR expert at all, but I looked here left and right. It looks like here, left and right. I have no minor state here. When we put the same RNA or DNA to a higher temperature, which is typically what's used to do these binding assays of Z-alpha to the nucleic acid, 42 degrees, then we start seeing that excited invisible state, right? Minor state, about 2% of the total population. So we have a minor state in the population of A. The question is that minor state Z or is it something else? And that's where more NMR comes in, shown here, where we superimpose here. So those are signals of the CH8 in purines, CH5 in CH6 in pyrimidines, and looking at, we have an RNA here that in purple peaks gives us, okay, that's an RNA with a methyl group at some position that gives us a control for a Z formation. Like it adopts the Z, those are the darker purple peaks. The one at the bottom is just the blue one, straight RNA, and that we melt at 70 degrees. So we increase the temperature to 70 to characterize that basically our minor state that I showed on the previous slide. If we look at all the differences here between blue and red and between the light purple, dark purple, and plot that on the graph that's shown on the right. So we see that basically our excited state, which are those gray peaks here, those gray circles, are better correlated to the melted state than they are to the Z conformation. So like the R square is like 0.97, and it goes down to like 0.6 or so for the Z. So what we saw on the previous slide, like, yes, we have an excited state in the population of A, is not Z. It's just something that's melted, basically. So we add another step, right, on that pathway now, where we have, okay, we know we can have sampling of A or B with the protein. We know we also have some transient duplex melting. And we showed after that we have Z-alpha binding to this, Z-like state formation. So we don't have the atomic details yet of what that Z-like state would look like. We're working on that, but this is somewhere here before we get to full Z-form stabilization and maybe propagation depending on the context. So people had shown that early on with bDNA, long stretches of CG repeats that, you'd bind cooperatively several Z-alpha domains. So we're in the process of, looking at that for RNA. So that's where we're at. So the conclusion here is that, if I go back to my main title, one step the right run, I don't know about the step and the run, but we have some right stuff that goes into left. And the spin in the middle, it's more like a melt, right? Somewhere in between the two, there's some kind of melting that leads to a Z-like conformation. And the protein is there to facilitate stabilization of the Z. How it does that exactly? We are working on it. No one knows in detail what goes on. It's still this is still the gap in knowledge right there. So Z alpha binds to this ARNA, right? We showed that. And that leads to the Z-like and then the Z. We know that, and we don't need high salt for this, right? So no detectable Z state among A population. And overall, so this Z-RNA thing, which was, controversial at first, the pathway from A to Z are relevant to biology, whether we like it or not, right? I think we have enough examples now. And I definitely encourage you to check out the abz.bio, which is the website we have associated with conference on everything non-A, non-B, so usually Z form, that we organize once a year online. And if you send us emails, we're happy to add you to the mailing list so you can check out the next conference. It's usually in the fall that we do it. And so this A-form helices, that kind of, we started thinking about that. I'm like, well, that's kind of boring because it's not where the motifs are. Well, it turned out to be maybe, if we understand them better, like these A to Z modules, once we understand, sequence context when that happens, particular environment, types of RNA, then maybe we have some A to Z modules that we can be used for engineering, for example. So I want to thank the people who have done the work. So we are in Houston. This is my lab. Parker is co-mentored with Beat. He is in Colorado, where most of this work has been done, working with Mokos and Jeff Kroll that I mentioned. We also have a collaboration with Mercedes. I didn't talk about this today. She is in an immunologist looking for, pretty much the connection of what we do to what's happening inside the cell in a cellular context. I mentioned Pascal in Strasbourg, and this was work that we did with Luigi and Philippe back then. I was in the Kieft lab for several years, which is now in New York. I mentioned Rob Welty, fantastic people leading core facilities, supporting us to get grants, the funding, obviously. And we're always interested in talking to people who are interested in this project, working on this in some fashion. We have Beat and I, this sort of, we used to be next door offices. Now we bring this collaboration to more, with more distance in between our two labs, but we keep working together. I have Nat for a postdoc right now that's up. You can check it out, send me an email, we can talk about it. Also welcoming graduate students. I'm on the graduate recruitment committee, so person you want to talk to if you're interested. We get plenty of undergrads here too. So, and thank you for listening, and I'm happy to take questions.
Thank you. There is already one from Reggie. I don't know if Reggie wants to read it.
Yeah, I can read it. Give me a second.
Hey, Reggie. Another Houstonian.
Another Houstonian. You got it. Yeah. Hi, although I'm not in Houston right now. I wish I was.
Yeah. You'll be here soon.
Yeah. Great talk, Quentin.
Thank you.
My question was, given that the Z alpha domains are in proteins like ADAR, which are involved in innate immunity, is there any evidence for Z-RNA being some kind of a pattern that's recognized by the innate immune pathway? Is it common in
that's
viral RNAs?
Well, absolutely. And this is what gave, I want to say, the biggest boost in confidence that this was biology relevant in the past 3 years. There's several papers in Nature that has come out, mostly around 2020 and since, that have shown that, even the people have shown that SARS coronavirus makes Z-RNA when it's inside cells, and influenza A virus, that there might be competition of the Z-RNA binders between the cellular RNAs, the endogenous RNAs, and the viral RNAs. So there's a huge field, and this is very cellular biology oriented. It depends mostly on antibody recognition to determine whether this is Z or not. So we don't have completely clear links yet on what is Z, what is not Z, but it's definitely relevant. I think now people have really characterized pretty well the double-stranded RNA sensing pathways that leads to the innate immune response and how Z-RNA plays a role in that. We have a small review on that Parker actually, we published that in RNA where we talk a bit about that.
Is there any sense if the Z-RNA is showing up in, there's these long double-stranded RNA segments that show up in a viral infection when the RNA is copying itself?
Yeah.
Is it there, or is it in the sort of the genomic RNA, the quote single-stranded RNA?
It's both. So it's both. It's the timing of that is not clear. What seems to happen is that well, I mean, the function, like the recognition, the presence of it is there. The recognition by the domain is like we understand that, but the function associated with what is recognized by the protein when and on which RNA and what does that lead to in terms of the response, this is not as clear cut, right?
I see.
So, but it is related to the innate immune response. It is related to the editing of the RNA because ADAR edits RNA. And the long isoform of ADAR, which has the Z-alpha domain, is the one that is hyperactivated when you have an innate immune response. So there's some additional editing that goes on that is tied to the presence of that Z-alpha domain. So we are trying to keep connecting the dots here as we go, and this is why I mentioned this immunologist collaborator that's involved in this, because we need that, right? But you're absolutely correct. This is exactly what's happening. This is where the field is going. And we talk a lot about this at this conference, ABZ meeting, that we call it. Yeah.
Thanks.
Rachel.
I have a question kind of about, I guess, the physical origin of the A to Z equilibrium. So what actually pushes that equilibrium? Specifically thinking about like how ionic composition certainly pushes it in some scenarios, but also how this protein also pushes it towards the Z. And I'm curious, the relationship between ion composition and protein, are they doing similar physical, is it a similar physical phenomena that's pushing that equilibrium, or is it very distinct? Is there experimental evidence for any of this?
Yeah, that's a good question. I mean, from what we see with the CD and all, it's a similar profile, right? It's the same. It's the same form of, we stabilize at least the same thing. The way to get there, that's less clear, right? Because until we know what that Z-like, I mean, there might be different Z-like conformations, like the conformation that would immediately precede the Z, right? Maybe very different depending on the ionic strength contest, the protein abundance, yeah. Good question.
So I put the link to the conference. So I have one question as well, but I guess it's impossible. Is there any way how these steps or motifs or modules, I think probably they should be named, could be predicted from the sequence and maybe context of secondary structure? There's
that's how I've been approaching this. And right now we're at a stage where we think it's a propensity thing. So we have what we've looked at also that I didn't talk about here is like, we have different rates of adoption for different sequence contexts. So some sequences work flip better than others, right? And that depends on what's around them too. Like these less stable A-form regions, maybe bubbles, maybe unclear. But that then it might be really broad. Right. So we are going for like genomic approaches to, can we map exactly where this Z-alpha domain binds on a population of RNA? So we're looking after this to look at trends at least. It may not be a sequence signature, but it may be a trend about certain contexts to at least rule out some contexts. It might not be enough that we can pinpoint for sure. What I was saying at the end, do we have an A to Z module Unclear. But that's where our thinking is going. I mean, we think in terms of biology, it may not matter as much. Like you want something, you want to fall, let's say you're in an A to Z flipping module, so you want to fall within a certain category, right? Either you're flipping or you're not flipping. Like I mentioned, some of these are like a lot of RNAs don't have a Z step anywhere. So it's almost like they made a point of not having a Z step, whereas some do. But we might fall in this, right? And at least we would be able to, if, designing an RNA, like an mRNA that goes in a cell, for example, like in a vaccine, right, that we want to send to patients, we could maybe have at least a system to give some confidence that it will or will not adopt Z in some regions, because that may mess up, as we just discussed with Riju, the immune response, right?
So I have one more related to that. So you showed that there are examples of various sequences that actually they can make the Z steps. But is there like some distributions of the sequences, like, or we just have to look more carefully for Z steps, and then pretty much we'll see that Z steps can be done by pretty much.
Yeah, that was, I mean, when we did that, there was definitely more of a bias, but that could just be because we don't have enough structures of Z steps. But it tends to be, I think UA and AA were pretty popular with RNA. So it's not, it seems to not be the same bias for DNA and for RNA.
Do we have more questions?
Can I quickly follow up on the bias that you just talked about? At least from what you've looked at, do you think that bias is driven by the energetics of the Z step itself, or just by kind of the energetics of what else that nucleotide could be involved in? So it's like, it's, I guess, is it that UA actually a stronger Z step, or is it just that UAs have weaker other interactions? So it's prefers to
Yeah,
go
so
into
we don't, I mean, we don't think this is, yeah, it's tricky to dissect what the contributions of all the aspects. I mean, we didn't think necessarily of that dinucleotide Z step as a motif more than an induced conformation by the stuff around it. And the stability, I mean, we have no clue. I don't think anyone has done that yet. I don't think Pascal has done it yet. Like the contributions of the stability, let's say if we had the same system and we put a UA, we put an AA, we put a CG to compare, right, at that Z step, we don't have a system that allows us to do that quite yet. Good question. Riju, you have another question?
Yeah, I was, you said right now the way that we can detect the Z steps, I mean, one way is through crystallography or high resolution cryo-EM. You mentioned that there's some antibodies that can also detect the steps. I was curious if there's any kind of chemical mapping signature for the steps.
So this is something that I mean, Houston, Tony Musto, Tony Musto will talk about it. We're talking about it of developing this. There was a paper, like Alex Rich published a paper in the 80s about mapping of the Z, because you do have, when you go from that right-handed to left-handed, the some positions on the bases are more exposed in the Z than they are on the A. And so thinking about harnessing that, it has not really been followed up, but I think now with the methods that we have, and all the various probes and we have in the toolbox, that's something we're going after, because we think we really need, this is one way to bridge the gap, right, between the antibody detection in cell and the high resolution crystal structure or NMR. Like we need something else that we can plug into a larger system than one can be looked at by NMR, or that gives us a reliable information. So that's what we're trying to build. Yeah.
Yeah, my guess is because the G goes to a SYN conformation.
Yep.
That'll change the reactivity of its, actually it was even if it's 2' hydroxyl.
Yeah.
So there could be a signature for shape reagents, and then both the N7 and the N1 position, their reactivities could shift. Actually, I think they could be suppressed. So, and Tony's developing ways to look at DMS reactivities of
that's
both
our
positions.
thinking. Yeah.
Yeah.
Yeah, we're moving in that direction. Absolutely. Yeah.
That's exciting.
All right. There's another question in the chat here. Yes, absolutely. But that's exact. I had one example, but that's what we use readily. If you look at our papers, we do use CD. That's our best. I want to say that's the first line of entry for monitoring A to Z is CD, circular dichroism. Before we do other things like ITC and MR. Yeah, absolutely. And that's traditionally what's been used also. I think that's how they worked in the 70s.
So then it should be possible to do some high throughput studies on various sequences and
Yeah, and we have done some of that. I mean, in our papers, we looked at several fragments that we're doing this. It's not always easy because when you get a mix, like if you get a fragment that, an RNA that completely shifts, then that gives a clear spectra. If you don't, because you get a combination or you get transients, it makes the spectrum more complicated to analyze. But we came up with a formula using the shifts at different wavelengths that enabled us to make sense of that. But it's not trivial. So, but it can be high throughput, absolutely, except that it's a bit costly to in material, right? Because you need a lot more for CD. Roughly how many examples of Z-RNA exist in the PDB at present? So in terms of Z steps, we have, I mentioned about 600 instances of Z steps. So that's in all these, larger RNAs. In terms of the Z-RNA double helix itself, there is one. So bound to Z alpha, the one I showed. There's one or two by NMR showing, I think one shows a dinucleotide or two base pairs, and they're from the 80s. So it's not that much. So we're definitely working on that too and thinking like, could we get a double-stranded Z-RNA with something else than CG and, a different context. So a lot of people are after that right now. Yeah. All right, well, thanks for your questions, everyone. Thanks for having me again.
Yeah, thanks for talking in our seminar. Great talk. I'm excited to have you. All right, I will close the session then. So thanks again, Quentin.
All right, see you next time. Yeah.
And this will be
hours.
uploaded. Bye, everyone.